Okay, good morning, everyone. Great to see a big audience for a science/immunology session. And we've got a real treat in store for you today. So this is our session about airway inflammation in CF, molecules and mechanisms. I'm Robert Gray from University of Glasgow in Scotland, not Glasgow, Glasgow, just in case anyone's wondering. And I've got my wonderful co-chair, Kristen Huddock, who is, I have to say, the best co-chair in the history of the Earth. In terms of how organised she is. Amazing. I don't think we need to be louder. Just need to listen. Right, okay. No session on inflammation is complete without Dave Nichols' so-called simplified view of airway inflammation. And I use this slide all the time, and lots of other people do as well. And suffice to say, we still don't know anything really about airway inflammation in CF. We're only just scratching the surface. But today you're going to hear some really great talks from some promising, young, developing academics who have brought the average age of the presenter down in this conference markedly today. So that's great. So we're going to talk about inflammation. And really, you know, you're going to find out a bit about what happens to lung disease in terms of inflammation before and after modulators. And our main objectives are to think about the types of immune cells involved, how they might affect inflammation, identify the impact of nature. So is the inflammation we see in CF due to CFTR mutations, or is it due to nurture? Is it due to the CF lung environment and infection? And then examine some novel approaches to the treatment of inflammation in CF and the scientific basis for these. So a great session, so I'm not going to say anything more. I'm going to go on to our first speaker. So our first speaker today is Tuba Kızılmırak, who is from Turkey, I believe, originally. One of my favorite countries, without a doubt. And she's going to come and speak to us today. Right, she's going to tell us about CFTR dysfunction and how this shapes airway immune cell compositions, contributing to lung pathogenesis in children with CF. And Tuba is a physician scientist who trained in Istanbul and is doing her second pulmonary fellowship at Yale, where she works with Dr. Clemente Brito. So really excited to see this data. We've been following the single cell story in terms of the sputum immune cell phenotyping for a while now, so this is going to be a great update. So, floor is yours.
Thank you. Hello, everyone. I'm Tuba Kızılmırak. I'm a pediatric pulmonary fellow at Yale, working also in dear Dr. Mary Egan's lab. And I want to start with many thanks to organizing committee and also chair of sessions for giving me this opportunity. And in this talk, I'll summarize some preliminary findings of our research focusing on airway immune response at an earlier stages of CF lung disease by utilizing single-cell transcriptomics. I'll try to use this. And I have nothing to disclose. So as we all know, airway inflammation is intense, excessive, sustained, and playing a central role in CF lung pathogenesis. So it starts with the lack of CFTR functioning at the apical membrane of the airway epithelium, which results in dehydration of the airway surface liquid, impairs mucociliary clearance, and causes mucus obstruction. And this creates an environment favoring chronic infections, resulting in chronic airway inflammation, which is characterized by excessive neutrophilic infiltration and excessive cytokine production, with contribution of the impaired immune response as a result of intrinsic CFTR defect in immune cells. This becomes a vicious cycle in CF lungs and sets a hyperinflammatory state and progresses into CF lung damage. In the meantime, it has been shown that macrophages and monocytes play a critical role in this process. They have an altered distribution in CF lungs that affecting many critical signaling pathways that are required to maintain lung tissue homeostasis. As a result, there is an imbalance between inflammatory response and anti-inflammatory response in CF lungs. So in 2020, Dr. Brito and his colleagues developed a novel, innovative approach to analyze immune cells present in the sputum of CF patients by utilizing single-cell transcriptomics for the first time. So why sputum? Sputum is a non-invasive and robust way, airway sampling, from the site of inflammation. Sputum also allows us to get information regarding response to the airway microenvironment, cell-to-cell, cell-to-pathogen interactions, as well as airway transmigration. So in this novel approach, they collected sputum samples from healthy controls and adult CF patients with a mean age of 35. The samples are processed to prepare single-cell suspensions and stored cryopreserved. At the time of assay, samples were thawed and utilized in a droplet-based single-cell RNA sequencing to get mRNA expression profile of the cells present in sputum. So in this study, they profiled a total of 20,000 cells from 9 CF patients and 5 healthy controls. In this figure, you are seeing a UMAP data. UMAP stands for Uniform Manifold Approximation and Projection. Here, I'd like you to think about a 3-dimensional count matrix, in which each column is data comes from each gene, and each row is data comes from each single cell. And the third dimension is data comes from each patient. And if you were to think, you can detect up to 20,000 genes from one single human cells, you really end up with high-dimensional data of sequencing. So UMAP is a dimensionality reduction technique that is used to visualize a clustering pattern of a high-dimensional data. So in UMAP, each dot represents each single cell. What happens? The cells sharing similar expression profile comes closer and creates clusters. And they are labeled based on known gene markers, and they are color-coded based on cell types. In this study, they identified 7 distinct immune cell populations present in adult cohort, including alveolar macrophages, neutrophils, T cells, natural killers, B cells, monocytes, monocytic macrophages, as well as dendritic cells. In this figure, you are seeing a UMAP data comparing CF patients to healthy controls, in which pink color is representing cells obtained from CF patients, and mint color is representing cells obtained from healthy controls. What they showed, there were significant differences in alveolar macrophages, neutrophils, and monocytes, monocytic macrophage cell groups, in which the majority of cells present in the sputum obtained from CF patients were neutrophils and monocytic macrophages, and a limited number of alveolar macrophages, less than 1%, present in their sputum. On the other hand, the majority of cells present in sputum obtained from healthy controls were alveolar macrophages and an abundance of or high percentages of classical monocytes present in their sputum, as well as a limited number of neutrophils were present in their sputum. So these findings were very intriguing and, of course, raised many additional questions. So are those changes observed in an adult cohort as a consequence of long-standing infection and inflammation in an adult population with advanced disease? Or are those changes intrinsic to the lack of CFTR functioning? If we were to evaluate a younger CF population, would airway immune cell profile be similar or different when compared to the adult cohort? And secondly, in the era of the CFTR modulator therapy, despite significant improvement in clinical care, there remains a knowledge gap, the impact of CFTR modulator therapy on immune response in CF patients. And if we were to evaluate a younger CF population who were on highly effective modulator therapy, I'm referring to Trikafta in this talk, would treatment with Trikafta change the airway immune cell profile? To address our questions, we've successfully implemented this innovative approach in younger CF population. And in this slide, I'd like you to walk me through to get better understanding the process of utilizing single-cell transcriptomics on sputum cells. So it starts with sample collection by sputum induction, and samples are processed to prepare single-cell suspensions. They are cryopreserved to keep the cells alive, and at the time of the library construction for sequencing, samples are thawed and undergo another processing to be ready for library construction. And during library construction, barcoded gel beads that each contains millions of barcoded oligonucleotides are mixed with single cells, enzymes, and reagents. They are squirted into the microfluidics at a flow rate designed to enable each gel bead would bind each single cell. They get immersed in partitioning oil and generate single cell gems. So they won't combine again, so you could get information from one single cell. Single cell gems are basically reaction vesicles. And what happens inside? Gel beads are lysed, RNAs are extracted from cells, reverse transcribed, and barcoded cDNA copies are produced. And barcoded cDNA copies in the next step pulled together, the oil is removed, and the cDNA copies are amplified to generate enough genomic data for sequencing. And following sequencing, there is a computational analysis of sequencing data. So you could identify cell populations present in sputum, as well as their characteristics. To date, we analyzed single-cell sequencing data from 10 patients with CF, age ranging from 12 to 25, with a mean age of 16 years. 6 of them were on Trikafta treatment, whereas 4 of them were not on any modulator therapy due to their underlying mutations that were not eligible for CFTR modulator therapy. And this cohort mostly had a preserved lung functioning with a mean % predicted FEV1 of 102, and with 2 exceptions in 2 patients showing mild obstruction in their lung functioning. And we profiled a total of 11,300 sputum cells present in their sputum. In this slide, we are seeing a UMAP data. Again, each color representing different cell types. We were able to identify 7 distinct immune cell types present in younger CF cohort. But what was interesting is that alveolar macrophages were present in younger CF cohort when compared to adult CF cohort, and showing a higher abundance in terms of percentages when compared to other cell types. But further, when we looked the cell distribution based on modulators therapy, what we showed that there were significant differences in neutrophils and alveolar macrophages. In this UMAP, pink color is representing cells obtained from patients who were on Trikafta, and navy color is representing cells present in sputum obtained from patients who were not on any modulator. Interestingly, the patients who were not on any modulator therapy showed increased number of the neutrophils and a limited number of the alveolar macrophages present in their sputum. In contrast, patients who were on Trikafta treatment showed abundance of the alveolar macrophages, whereas a limited number of the neutrophils. Moreover, regarding monocytes and monocyte macrophage cluster, we identified 4 distinct subclusters of monocyte macrophages, in which 3 of them showed increased expression of genes that are associated with inflammation resolution, cell survival, and host defense, such as MSR1, ApoC1, C1QB. On the other hand, 1 cluster showed increased expression of genes that are associated with inflammation, such as IL-1 beta, CXCL2, 3, 8, CCL3. But what was more interesting that when we looked further the distribution based on ETI treatment, the 3 subclusters showing increased expression profile of anti-inflammatory signaling were obtained from patients who were on Trikafta. In contrast, the cluster showing increased expression of inflammatory signaling were overrepresented in patients who were not on any modulator therapy. But also, this cluster was still present in patients who were on ETI treatment. In conclusion, alveolar macrophages were present in sputum immune cell profiles of the younger CF cohort when they were on ETI treatment. However, patients not on any CFTR modulator therapy showed similar pro-inflammatory sputum immune cell profile as previously reported in adult cohort. And in addition, the monocyte-derived macrophage subclusters showed distinct differences in their gene expression profile based on ETI treatment. So this is an ongoing research, and we'll continue to expand sample size by adding more samples, now aged down to 6, to enhance the generalizability of these findings. So we will have a trajectory from early childhood to young adulthood. And there's an ongoing validation studies to refine these findings and ensure the reliability and accuracy at a protein level. With that, I want to thank to all people you're seeing on the slide that have helped me to make this happen. I want to especially thank to my dear commenters, Dr. Burrito for his expertise in single-cell sequencing, dear Dr. Borussia for her invaluable support throughout the project, and dear Dr. Marie Egan, well, actually for everything, for standing behind me through every step of the way, including my visa issues. And, and Caitlin and Hill for their help during computational analysis piece. My funding sources, I can't thank you enough, Cystic Fibrosis Foundation, Pediatric Scientist Development Program that allowed me to thrive as a physician scientist, as well as their endless support, Yale Department of Pediatrics. And with that, happy to have your questions, and please visit my poster to number 219. Promise it's gonna be easier to find me there.
Well, one excellent talk. We'll take some questions from the floor now, if there are any.
So what happens to our alveolar macrophages? Where do they go? Why aren't they in the sputum of CF patients?
So, again, my argument is, or my hypothesis is, due to the intrinsic CFTR defect in immune cells, due to the abnormal signaling, there is a shift from alveolar macrophages, which will be present in a healthy patient, is making a shift towards from alveolar macrophages to an abundance of more pathologic or inflammatory inflammatory cell populations, such as monocyte-derived macrophages, as a result of recruitment into the lungs.
But do you see that shift? I mean, do you see that shift to a different cell population? I mean, I know you see the monocytes coming in, but the alveolar macrophages themselves, what do you think happens to them?
So the source of the alveolar macrophage is also monocytes. They are recruited from the peripheral blood. And I think there's a difference in terms of proliferation of the monocytes and differentiation into the different cell types. And that's what we are seeing. Instead of differentiating into the macrophage profile, they chose to, as a result of the, again, pro-inflammatory signaling, they are differentiating into the inflammatory subtype.
So you don't think there's a difficulty of those macrophages to make it into the sputum?
So I don't think it's a technical issue, because if they are there, we were able to see them in the sputum with a higher abundance, like 80–90%.
Okay.
Yes.
Hi, Judy Voinow from Children's Hospital of Richmond. So, very nice presentation.
Thank you.
I would like to know, what was the sputum microbiology like? So could some of your results be due to shifts in the microbiome?
That's a perfect point. So in this young cohort, we don't have any patient colonized with Pseudomonas. They are showing still colonization of the staph virus, mostly MSSA and Periphelis hemainfluenza. Haemophilus parainfluenza, okay? Well, it's difficult to say with 100% of sure, you know, there's definitely a contribution of the colonization.
Yeah,
I agree.
I think that would be interesting to look at as well, in addition to the...
Yes, fully recognizing the sample size here, I couldn't make the subgroup analysis. It will be great to see. Thank you.
Thank you for that talk. I think that the new cohort is so interesting. And getting back to this alveolar versus recruited macrophage, in terms of how the single cell, which you went through very beautifully, works, is the numbers you're showing us, is that proportions of cells or total numbers of cells? And so in terms of the people not on modulators, are they, and getting to like what's coming out of their lungs versus the patients on modulators? Like, do we know if they have bronchiectasis? Are they, and when you look at their sputum, is it a proportion where you're getting? So two questions. Is that a proportion or an absolute number, what you were showing us when you say less alveolar macrophages? And then what do we know about the health of the people not on modulators versus those who are on modulators?
So when you visualize your data by using UMAP, it uses counts of the cells from each patient. It's not proportion. Okay. But when I further looked at the proportion, there is a difference between the proportion of the alveolar macrophages in patients who are not on any modulator versus on Trikafta, for sure. High differences like 5 to 7% versus 80s. And what was the second question?
So in terms of health, I'm just wondering in terms of if it's 10% alveolar macrophages, is that because you're just getting so much more, mucopurulent material and recruited cells because they're sicker, that the alveolar macrophages are in their lungs, just that what they brought up is a higher percentage of recruited cells?
So regarding that point, I don't think these are kind of reflecting the technical issues here. If they are there, you're getting the result, the data. But it's a great point. But my argument will be the most vulnerable cell types in the sputum is neutrophils. I wouldn't see them if that's going to be the case. So alveolar macrophages is kind of more stable. So if they would be there, I'm assuming that I would have the data from the alveolar macrophages. And if that's going to be the technical issue to see the alveolar macrophages, I wouldn't see, because we've been kind of uploading the microchips at the same time. They were treated evenly. So, yeah.
So we've got time for one really, really short last question, just for the sake of giving all our speakers a chance to have their presentations.
Erica Orsini from the Cleveland Clinic. Fabulous talk. I was just wondering, we study macrophage mechanobiology, and we've often used these data sets to try to look at expression of mechanosensitive ion channels, but we notice that often these genes are missing. Do you know why that would be, that certain genes are not getting picked up in these data sets?
If I understood your question, so we are not necessarily picking up the cells, gene expression profile of the ion channels?
Yes, the mechanosensitive ion channels in particular.
I haven't specifically looked at, sorry, I don't know yet, but definitely happy to take a look.
Okay, thank
Thanks
you so much.
for contribution.
Okay, thank you so much.
Our next speaker is Dipali Luthra, who got her PhD in Oklahoma State and is a postdoc at Emory in Dr. Rabin Turovanzium's lab. I'm sorry, Rabin. Anyway, her goal is to understand CF lung disease driven by neutrophilic inflammation through an immunologist lens. The title of her talk today is actually changed, and it's going to be Regulation of Secreted Mediators by Human Neutrophils Adapted to the CF Lung Microenvironment.
Thank you so much. Thank you, everybody, for being here. I'm Dipali, postdoctoral fellow at the Turovanzium lab at Emory. I would like to start my talk by first thanking the NACFC organization, NACFC and CFF, for giving me this opportunity, and also to both our lovely chairs. So today I'll be talking about regulation of secreted mediators by human neutrophils adapted to the CF lung microenvironment. I have no disclosures. And I like to start my presentation every time by a picture of Olivia. This is the first report that I ever read about a girl, a 9-year-old girl suffering with CF, which really got me hooked to this field. And I think one thing we all can appreciate is what binds us together today in this room is our drive and passion to understand CF and also make their lives better. One of the most common clinical manifestations of CF is the airway lung disease. And we now know that the healthy airways have a thin layer of mucus. The pathogens are inhaled in, and neutrophils and macrophages are always there to clear any infection. Whereas in CF lungs, there's a lot of muco-obstructive disease because of a triad of mucus plugging, influx of neutrophils, macrophages being there, also a lot of bacteria and fungus causing infections. Now, all of this leads, along with the unknown stressors that are there and local hypoxia, leads to not just neutrophil and macrophage-associated inflammation, but also bacterial infections, leading to permanent lung damage or structural damage. Something that our lab has been studying from last 15 years or so is that neutrophils in CF adopt a very specific pathological fate. They are not only alive and non-apoptotic, they are also metabolically licensed. They hyperexocytose primary granules and also promote the influx of neutrophils into the airways. And these neutrophils are actively repressing bacterial killing by de novo synthesis. To this, we have coined the term as GRIM, which stands for granule-releasing immunomodulatory and metabolically licensed neutrophil. So for the rest of the talk, if I mention GRIMs, that means I'm talking about CF neutrophils. So in the CF airways, there's basically in this triad, there's a stable pathological environment that is being created. And neutrophils, being the main protagonists here, they exert primary control over other immune cells, such as T cells and macrophages, by downregulating the macrophage scavenger receptors that are causing inflammation. At the same time, they're also releasing neutrophil elastase, myeloperoxidase, methionine sulfoxide, and so many other cytokines and chemokines and factors, which are not only establishing ties with lung epithelial cells, but also microbial species, as well as they're transcriptionally repressing the bacterial killing at the same time. Now, these neutrophils, we know that are certainly secreting a lot of chemokines, cytokines into the system, into this microenvironment. And the state of the art is such that from BAL and sputum samples, we now know that there is high IL-6, IL-8, IL-1 beta, and TNF-alpha present in these two biofluids from CF, whereas IL-10 and interferon gamma is downregulated. There are also some changes in the epithelial cell and monocyte macrophage secretome that have been described in the literature so far. But what we do not know is how are these GRIMs contributing to the cytokine microenvironment? And that's the focus of my study for this study. So to understand this better, we have isolated the contribution of just neutrophils to this microenvironment, for which we get blood from CF and healthy donors, isolate the neutrophils from both of those, and transmigrate them through an Alvatex transmigration epithelial model that we have in our lab, in which neutrophils are transmigrated towards LTB4 and CFASN. Now, LTB4 is the leukotriene-4, which is a chemoattractant for neutrophils that we add into the system to obtain the healthy neutrophil. And CFASN is the CF airway supernatant, which is basically purified from CF sputum, obtained from CF donors as well. Now, upon exposure to CFASN and transmigration through this model, we now know that we are able to create a healthy versus CF phenotype in our lab, which has been published by some of the studies from our lab and other groups. And these GRIMs, or these CF neutrophils, are not, as I mentioned in the previous slide, they're not only alive, but they're also metabolically licensed, they hyperexocytose primary granules, and are also repressing the bacterial killing. Now, at this stage, we know that post-transmigration, these neutrophils are certainly releasing some cytokines and chemokines or secretory mediators in this microenvironment. Also, if we condition or if we let them condition some media for 24 hours, they will specifically, just post-washing, they themselves will also be releasing some cytokines and chemokines. So we have split this study into 2 phases. Phase 1, right post-transmigration, and phase 2, post letting the media, letting these neutrophils condition the media and using the supernatant to analyze the profile. And that was done using the electrochemiluminescence assay technology by mesoscale discovery. So for this study, we have 8 healthy donors and 6 CF donors. And I will be splitting the data into 2 different sets for ease of understanding, because this is still our pilot understanding of this study. So in the beginning, I will be talking about the cytokine profile or the secretome coming from 2 healthy subjects, and eventually I'll walk you through the other validation data set as well. Now, in our first pilot study, we found out that the pro-neutrophilic markers, such as IL-6, IL-8, are upregulated, as well as pro-monocytic markers like MCP1, MCSF, and this certainly is suggesting that TNF-alpha as well, this certainly is suggesting that there is a upregulation of some of the pro-monocytic and neutrophilic markers in CFASN recruited neutrophils. The red ones here are the CFASN recruited neutrophils, or the GRIMs, and the blue suggests the healthy neutrophil. And these are coming from 2 healthy donors. When we went on to study some of the inflammasome and interferon markers, we saw that there is almost inverse regulation of inflammasome and interferon secretion. As you can see here, IL-1 alpha, 1 beta, and IL-1RA are upregulated in this system, whereas all the interferon markers, IP10, interferon gamma, ITAC, and IL-29 are downregulated. There is a study that was published from our group a few years ago, which suggests a feed-forward inflammasome signaling by some of the extracellular vesicles that are present in the CFASN. And we have recruited these, transmigrated these neutrophils towards CFASN. So that kind of made sense. When we went on exploring this data set further, this clustergram suggests that you can clearly see that there is a upregulation of IL-1 alpha, beta in CFASN recruited neutrophils, whereas downregulation of interferon markers such as IP10. Now that was very interesting and something to be chased. However, when we added 6 more CF donors and 6 healthy donors data set to this, we saw that one big thing that stands out right off the bat is all of the markers are showing no significant difference. So basically, there is no donor-specific difference in the cytokine or the secretome secretion by these subjects, by the neutrophils from these subjects. Here in this graph, you can see that the blue and blue represents the neutrophils transmigrated in LTB4, whereas the red ones are transmigrated towards CFASN. The first 2 represent healthy donor, and the third and the fourth box bar represent the CF donors. Now here, one thing that is certain, that there is no donor-specific difference. However, the IL-6 and IL-8 seem to be very saturated. So this suggests that we need a rerun. This data set was literally obtained last week, so we are still in the very beginning of understanding this whole profile. When we checked the pro-monocytic markers, we did see a significant difference in MIP1 beta in between the CFASN recruited neutrophils from healthy and CF donors. So basically the second and the fourth bar here. When we went on to further investigate inflammasome and interferon markers, again, similar thing that there's no donor-specific difference. Just visually trying to understand this, there's no donor-specific difference if we compare the blue to blue and the red to red here. However, one thing that did stand out was VEGF, which I'll talk about in a minute. So here again, you can see that some of the markers that we were very interested in, which was like IP10 and IL-1 beta, both of IL-1 beta is saturated and IP10 is very low. So this probably demands a rerun or reassessment of this entire data set, like by diluting some of the analytes for IL-1 beta analysis and also concentrating for IP10 to bring them all to scale. Now, when we took this entire data set, we now know from what this data suggests is that healthy and CF donors are not different across the board in the cytokines that are measured, except MIP-1 beta. So we ran a paired statistics. We combined everything together, LTB4 and CFASM conditions, ran a paired statistics, and one thing that did stand out very much is VEGF. VEGF was significantly different when compared between LTB4 and CFASM conditions. As you can see here, there's almost a twofold difference in between both of them. And also when paired statistics, including Wilcoxon, was ran, we saw that the p-value was almost 0.038. which suggests there's a significant difference there. Now, VEGF is an angiogenesis factor, and we know from some of our previous studies or previous analyses done in different studies from the lab, is that there is probably some role of VEGF in the airway lumen, possibly in the myeloid to myeloid signaling or cell-cell signaling. So this leads us to a very interesting stage that we need to explore this further. So the key takeaways here are that there's no donor-specific genotype difference on the secretome of transmigrated neutrophils, and similar to all the other phenotypes that we have seen by GRIMS in the past, the 4 studies that I showed which have been published suggesting that they are GRIM. So that's great. And second was in the validation data set, we saw that there is a saturation of IL-8, IL-1 beta, and also very diluted IP-10, which certainly demands a rerun for the phase 2 of the study. Additionally, if you remember from the slide where I showed phase 1 and phase 2, we really need to go back and see what phase 1 of the analysis, phase 1 of the data is suggesting, which is right after transmigration of these neutrophils before letting them condition the media. So that and also we want to investigate if the supernatant in the EVs in the supernatant or extracellular vesicles in the supernatant post-transmigration are really leading to any changes in these cytokine release or not. So all of this is currently still we still have an open question, which is that is there an alteration in the mediator secretion contributing to this chronic dysfunction of neutrophils, and is it because of inflammasome, interferon, or some other pathways that are being hit? So there's a lot more to do, and we are just getting started. With that, I would like to thank my mentor, Dr. Thiruvanziyam, for his commendable, in caps, commendable mentorship and guidance, and all of the team that we have at Emory Pediatrics, including all the labs. Also CF Atlanta and CF Scholars Program for giving us a space as early researchers to really come and connect with everybody and understand CF from different perspectives. Our funding agencies, NIH and CFF, and also everybody who's here. Thank you so much.
Thank you. That was a great talk. Questions? I'm going to ask one really quickly before the first audience member.
Sure.
So what is it about transmigration that you think changes the neutrophils? Is it contact with junctional proteins? Do you have any idea sort of what would change that phenotype?
I am not exactly sure what I cannot point one thing that can certainly lead to this, but we know that just the There are extracellular vesicles which are present in post-transmigration. In the supernatant, there are some extracellular vesicles which are present in the supernatant, which really have some, those surface markers do suggest that they are very closer to the CF neutrophils. There's a whole project in our lab, which is on poster 199, and the paper for it is currently underway. That does explain a little bit of what we understand from, yeah.
Okay, thank you.
Thank you very much. I've got a technical question. You said that you use ASN as a way to get the neutrophils through, right? And you said it's well to elicit the neutrophil chemotaxis. So, and you said it's a sputum purified. So is it just dithiostratol, or is it more elaborate way of purification? My question is whether what's the percentage, if you want, of the contribution to bacterial products in there as opposed to host-derived products?
Great. Good question. Very good question. So what we do is we take the sputum from CF patients. We dissociate it with PBS-EDTA to get a purified CF airway supernatant. That, to answer the first part, that does act as a chemotractant for the neutrophils in the transmigration model. But during purification, we are doing spins to kind of remove all the epithelial cells or the bacterial cells and all of that from the sputum, from the crude sputum. However, this CF-ASN does contain a lot of metabolites, some of the cytokines, chemokines, secondary mediators. A lot of that is still there. So this is basically our way to create a more physiological relevant environment in vitro in the lab. And the studies that have been published do suggest that post-transmigration through this model and exposure to CF-ASN does indeed lead to a CF-like phenotype. And there is like no difference between the GRIMs that we obtain in our lab from CF, from healthy neutrophils versus the CF neutrophils.
Yeah, the ASN are not filtered afterwards to remove bacterial. So you just have bacterial products like, say, LPS and stuff like that, but you don't. Okay.
Yeah, and we do dilute it to a certain stage so that and later on, the reason I was showing you the phase 2 of the analysis, because post-washing and all of that, we are actually removing all the CF-ASN recruited cytokines to kind of not have them mess up with our analysis.
Thank you.
Yeah.
I'm Sahana Krishna from the University of Pittsburgh. When you were talking about GRIMs, the fact that they're non-apoptotic, they're recruiting other inflammatory neutrophils, they also don't function as well as normal neutrophils do. This sounds an awful lot like senescence in that phenotype. Have you,
like what?
Like senescence and the senescent phenotype.
Okay.
Have you looked into that, into kind of just this phenotype of senescent neutrophils and how that might be playing a role in CF?
No, we haven't, but
I was just curious.
yeah, something, food for thought. Yeah, thank you.
Great talk, Emanuela Bruscia, Yale University. So you have a very interesting system in which you can answer a lot of questions, mechanistic questions. I'm wondering whether you have, so you are collecting sputum, supernatant sputum material, and I'm wondering whether if you have ever stratified those sputum material based on the age of the patients and the disease progression. So is this something that you, if you take sputum from children, would you see the same GRIM phenotype in your neutrophil, or that phenotype would be much milder? I'm wondering if that is the perfect system in which you can start to ask those questions.
So there are some studies that have already been published from our team a couple years ago, where the BAL and sputum was taken from CF subjects, from children, adults, adolescents, and then some of the studies do suggest that there is a differential expression of all these markers. And coming back to your GRIM question, that if they are GRIM as well, yes, they are, from all different age or stage of the disease progression.
Hi, I'm Anna Pistocchi from the University of Milan. I was wondering if you want to check also the microRNA present in the EV extracellular vesicles of neutrophils, or EVs derived from other type of cells.
So poster 197, that's another study from our lab. The RNA in the EVs of neutrophils recruited in CFASN and also healthy, the comparison study and all the transcriptional analysis that has been done. We have great data set. Please visit poster 197. Thank you.
Hi, I'm Megan Hirsch from UAB, and great talk. I just had a question. We're actually trying to recapitulate this phenotype in our lab as well.
Nice.
And I've seen a huge difference between the recruited levels with and without the CF sputum. So is that something that you've also run into with like decreased levels of recruitment from the LTB4 comparatively to the sputum?
So like post-transmigration, actual numbers are low?
Yes.
So there's a very slight difference, but there is. In CFASN recruited neutrophils, the count is less. The viability is also slightly less. It's not significantly different, but there is like a repetitive trend of lesser CFASN recruited neutrophils to LTB4 recruited neutrophils.
Awesome, thank you.
Yeah, thank you.
Thank you for the great questions.
Okay, a great interactive session so far. So next we have Domenico Matosco, who's an assistant professor from the University of Chieti in Italy. And just a point before you start, that'd be a brilliant place for an invited meeting on inflammation,
Yes.
having looked at where it is on the map. So just to put that out there. He's going to talk to us, however, about targeting platelet activation with pro-resolving mediators, an innovative strategy to dampen lung inflammation in CF. Floor is yours.
Thank you. Thank you for this kind introduction. Of course, thanks to the chairs for giving me the opportunity to talk about our work. So I have nothing to disclose. As we know, airway inflammation, dominated by the massive presence of neutrophils, is still the primary cause of health problems in CF. But despite that, the current anti-inflammatory treatments, such as ibuprofen, despite providing some clinical benefits, are still limited in their use due to long-term side effects. And in addition, more recent data show that although highly effective modulator therapies could lead to significant reduction in some inflammatory parameters, others are not affected, thus suggesting that to date, novel therapeutic approach to treat inflammation are still urgently needed. Over the course of the years, we now know that in addition to neutrophils, other cells may play a significant role in sustaining chronic inflammation in CF airways, including epithelial cells, monocytes and macrophages, endothelial cells, but also platelets. And our groups become interested in the role of platelets in CF since we demonstrated several years ago that people with CF express elevated markers of platelet activation, such as thromboxane B2 or membrane expression of P-selectin, that again are markers of platelet activation. And these findings in people with CF were reproduced also in mouse models in which we can observe an increased production of thromboxane B2 in gut-corrected CFTR KO mice, but also in Delta F508 mice. And importantly, we can also observe an increased expression of P-selectin in CFTR KO mice again, but also in this newly generated mouse model in which CFTR has been depleted only in platelet that has been named CFPF4 by Mark Looney in San Francisco that generated these mice. So collectively, these results suggest that the basic CFTR defect is responsible for platelet activation. And this hyperactive platelet sustain chronic inflammation while dampening beneficial pro-resolution response, mainly by affecting the activity of leukocytes that are aggregated to platelets, in particular neutrophils. So platelets sustain inflammation by modifying the activity of neutrophils in platelet-neutrophils aggregates. And importantly, what we also observed several years ago is that the increased platelet activation, as measured by the increased thromboxane B2 levels, is associated with worsened pulmonary function, as you can see here by this negative correlation, which you can see that patients with the higher platelet activation also show the reduced FEV1. also reduces lung respiratory function. Therefore, we can conclude for all these experiments that the basic CFTR defects skew platelets to a hyperactive, pro-inflammatory, and low resolving state that sustain chronic inflammation and impairs lung function. However, recent data that we obtained in our lab challenge this view in which the basic CFTR defects is responsible of platelet hyperactivation, since, as you can see in this graph, we found that in our people with CF treated with ETI, we did not observe a significant reduction of platelet activation as measured by thromboxane B2 release over time, thus suggesting that in addition that the restoration of a normal, almost normal function of CFTR is not sufficient to abolish platelet hyperactivation. And this is also consistent with our whole data showing that if we treat platelet with a specific CFTR inhibitor, again, we do not observe an increase in platelet activation. And again, we also see now in mouse models in which CFTR has been depleted just in the platelet lineage, that again, we do not have an increase in thromboxane B2 production. So collectively, these results suggest that in addition to the basic CFTR defect, something else is needed to promote platelet hyperactivation. Therefore, to study the contribution of airway inflammation in platelet activation, we took advantage of this mouse model, the beta-NAC overexpressing mice that has been generated by Professor Moll in Germany. And this mice is able to recapitulate a number of lung effects present also in CF patients, such as mucus plugging and neutrophils infiltration in absence of CFTR mutation. So here we have a sterile inflammation without CFTR mutation. And also in these mice, we observe a consistent increasing marker of platelet activation, such as thromboxane B2, platelet neutrophils aggregate, and also we observe an increase in platelet membrane p-selectin expression. Again, suggesting that maybe the inflammatory CF-like inflammatory environment is sustaining platelet hyperactivation. And also, also very importantly, we found that neutrophils in these mice release neutrophil extracellular traps only when they are bound to platelet. And this effect, this release is further increased in beta-NAC mice, corroborating that platelets are indeed a key drivers of lung inflammation. So based on these results, we hypothesized that platelets are hyperactive in CF due to both CFTR dysfunction and CF inflammation. And this is our working model in which we believe that CFTR dysfunction prime platelet to overreact in presence of CF inflammation to further exacerbate the platelet hyperactivation that drives finally lung pathology. And all these states are marked by p-selectin and thromboxane B2 expression, elevated expression of p-selectin and thromboxane B2. And in particular, we believe that elevated thromboxane B2 expression is a marker of platelet hyperactivation due to inflammation. So, based on this model, the aim of our work is try to revert this hyperactive platelet phenotype by using a family of compounds named resolvins that stimulate the resolution of inflammation. So first of all, we try to understand whether resolving could indeed reduce platelet activation, and to do that, we performed this in vitro assay reported here, in which we purified platelet from CF blood, treated with resolvins, with a number of resolvins in presence of some canonical inflammatory stimulus, such as arachidonic acid or Pseudomonas aeruginosa. And this, as you can see from this graph, among the resolvins, we found that resolvin D3 in particular, but also resolvin E1, are able to reduce platelet activation measured by thromboxane release. And these two, resolvin D3 and E1, were also able to reduce platelet activation in presence of a bacterial challenge, and also reduces thromboxane production by platelet in absence of any stimulus, so in resting platelets. Quite surprisingly, when we repeat the same experiment, purified platelet exposed to resolvins, but when we look at p-selectin expression, we found that neither resolvin D3 or resolvin E1 were able to reduce the overexpression, the platelet p-selectin expression on membrane, thus suggesting that resolvin D3 and E1 selectively attenuate thromboxane production, modifying specific platelet activation pathways. Again, we also repeated the same experiment in presence of healthy platelets, and as you can see from this graph, what we found is that resolvin D3 or E1 failed to reduce thromboxane production in both healthy platelets or in healthy platelets treated with CFTR-specific inhibitor. Therefore, collectively, to us, these results suggest that resolvin D3 and E1 selectively reduce platelet activation caused by CFTR-dependent inflammation that is not due to CFTR mutation. In line with this hypothesis, we found that resolvin D3, in particular, we moved to an in vivo environment where we found that resolvin D3 is unable to reduce thromboxane release in mice, CFPF4 mice, so in mice where CFTR has been depleted only in the platelet lineage, as well as resolvin D3 is unable to reduce thromboxane production, so platelet activation, in mice with CF-like inflammation. Opposite, what we found is that resolvin D3 was able to reduce thromboxane B2 release, so platelet activation, in CFTR KO mice after a bacterial challenge. Therefore, collectively, we believe that these results indicate that resolvin D3 is able to reduce platelet activation due both to CF-like inflammation and to CFTR defects in multiple cell types. To understand whether this reduction in thromboxane B2 could be relevant for lung pathology, we performed some correlation analysis by measuring levels of thromboxane B2 and neutrophils, or the total bacterial load, in mice challenged with Pseudomonas aeruginosa. And as you can see from this graph here, this positive correlation indeed suggests that the increased thromboxane B2 production is relevant for lung pathology because mice with the higher thromboxane B2 also are infiltrated with increased number of neutrophils in Airway and are also characterized by an increased bacterial load. Then to test resolvin D3 bioaction, we again repeated the same in vivo experiment with acute challenge with Pseudomonas aeruginosa, treatment with a single dose of resolvin D3 3 hours later, and sacrifice the day after. And in this condition, what we found is that resolvin D3 is able to significantly reduce total bacterial load, leukocyte, and in particular neutrophils number only in mice in CFTR KO mice, again consistent with our hypothesis that resolvin D3 is just able to reduce platelet activation in this condition. And these results to us suggest that resolvin D3 is stimulating the resolution of inflammation. Consistent with this hypothesis, we also found a reduced expression in mice treated with resolvin D3 of a couple of cytokines involved in immune stimulation. Again, to corroborate the hypothesis that resolvin D3 is stimulating resolution of inflammation, we performed a lipidomic analysis on lungs of mice infected, as above described, so with Pseudomonas aeruginosa, and sacrificed the day after. So as you can see from this, and treated with resolvin or vehicle, of course. As you can see from this heat map, but I guess more clearly, as you can appreciate by the representative histograms reported here, what we found initially is that the absence of CFTR is linked to a reduced production of pro-resolving mediators, including lipoxin A4, as you can see here, thus suggesting that the basic CFTR defect is associated with a poorer resolution of inflammation. However, we also found that treatment with resolvin D3 is unable to restore levels of pro-resolving mediators, but opposite, what we can clearly see in this graph here is that resolving D3 reduces the production of pro-inflammatory mediators, including prostaglandin E2, but also other pro-inflammatory mediators that are listed here in the heat map. So in the end, what I'm trying to say is that our treatment with resolving D3 restore the altered balance between pro-inflammatory and pro-resolutive lipid mediators only that we observed in KO mice, thus shifting the inflammatory response in CFTR KO mice toward resolution, as you can appreciate by the principal component analysis reported here. To analyze whether all these effects of resolving D3 were dependent on platelet, we again repeated the same in vivo experiment of Pseudomonas aeruginosa challenge, but before the challenge we treated platelets with a specific antiplatelet antibody that works very well, since as you can see from this flow cytometric representative dot plot, platelets are almost completely depleted by the antibody in blood, but not in lung. So in this condition, in absence of circulating platelet, we observed that resolving D3 failed to reduce thromboxane B2, to reduce total bacteria, and to reduce neutrophils infiltration, and also failed to reduce the production of pro-inflammatory mediators. Therefore, suggesting that resolving D3 alters circulating platelet in some way to promote resolution of lung inflammation. To further corroborate this hypothesis, so that resolving D3 modify platelet and platelets alter the inflammatory response against bacterial challenge, we also ran this in vitro experiment using cells isolated by CF donors. And as you can see here in this graph, what we found is that resolving D3 is able to to control bacterial growth only when neutrophils were co-incubated with platelets, maybe because this reduces bacterial growth could be due to an increased internalization of the bacteria by neutrophils, but again, only when platelets were present. So again, corroborating the hypothesis that resolving D3 acts through platelet to alter the inflammatory response against Pseudomonas. So in summary, we build this model here, in which we believe that the resolving D3 treatment, by affecting platelet in circulation, reduces platelet activation, blood platelet activation, and this in turn modify the action of leukocytes that aggregates to platelet, including neutrophils. This reduces platelet activation, then leads to let's say, a less pro-inflammatory and more pro-resolutive neutrophils that migrates into the lung, where it's more able to control bacterial growth together with platelet in a pro-resolutive environment. Finally, coming back to our first model, we believe that resolving D3 is therefore able to reduce platelet activation due to both CFTR defect and CF inflammation, restoring a less hyperactive phenotype that drive a better resolution of CF inflammation. Therefore, we are really convinced that targeting pro-inflammatory platelet in CF may provide clinical benefit. Let me finish my presentation with the acknowledgement. I would like to thank all people involved in this work, in particular Simona, that is our talented PhD student that is leading this project, and Matteo, that is here with me for his huge work with all the lipidomic analysis. Of course, thanks to our collaborator and to our funding agency, but in particular to the Italian Cystic Fibrosis Foundation that fully support our studies on platelet and CF. I will be happy to take your questions, but if you want to know more about this and other projects that we are running in the lab, please came to see our posters here. And thank you for your attention.
So what a great talk with a lot of data. Can I ask one question first? Sure. It's really simple question. So in CF, do you have an increased number of platelets as well as increased activation? Or is it the same number of platelets as some without CF?
So the number is the same. It's just a matter of activation. Okay. So you saw just the increased thromboxane and increased P-selectin, but the number is actually the same.
Okay, great. Ben?
Great talk.
Thank you.
Do you think there would be any additional benefit from anti-aggregation factors like p-selectin monoclonals?
Benefit for what, sorry?
Like a p-selectin monoclonal, like
could be something like we will try to look, because p-selectin is the protein that mediates the aggregation among platelets and neutrophils, and also with other cell monocytes or leukocytes. So maybe this strategy could work, because if we inhibit or let's say reduce the hyperactive platelet binding to neutrophils or to whatever it does, we could reduce the activation of the following leukocyte. Yeah, could be a good idea. Yeah, thank you.
Can I just ask a question? I really enjoyed your talk because I don't think we talk about platelets enough. One question I have is, I think aspirin inhibits thromboxane too, and I think the pro-resolvin mediators are super cool, but we have great data with aspirin. Is there a role for aspirin in this?
I was hoping in this question, actually.
Okay.
Yeah, we have a drug that reduces thromboxane that really works very well. So yeah, why not? I don't know. There are few reports that analyze the use of aspirin in CF. Maybe because I'm not a clinician, of course. Maybe because clinicians worried about side effects of aspirin. I mean bleeding, for example. But in my opinion, my biological opinion, I mean, I'm not a clinician again. Aspirin could have some side effects, but what are the benefits? So if the benefits are higher than the side effects, so why don't we can just try? The aspirin, so in addition to the side effect, probably is also not used in CF because an old paper showed that aspirin, but also ibuprofen could inhibit CFTR expression. So maybe some people could be worried by this effect, but this effect has been demonstrated in a cell line, so I don't know if it's really, really true. And in addition, I don't know. If I were a clinician, I would try to give some low-dose aspirin and see what happens. Also because low-dose aspirin seems to be safe, as demonstrated by the colorectal cancer screening program. So why not?
Yeah. Thank you.
So my question for you is, there are some, in your model, you think that your resolvins are acting on the platelets, on the circulations, and this is the mechanism of actions. But there are some data from Luning, actually, suggesting that in the lungs, there are megakaryocytes that are residing there and that they are producing platelets that may be very important during the process of inflammations. So do you think that in your model, you can kind of understand this is local, or if it's really about the circulation?
Yeah, sure. I of course knew about the megakaryocyte platelet production that occurs in lung. Maybe this is one of the reasons why we did not observe any depletion of platelet in lungs when we treated with the antibody. I don't know, or maybe it's just a matter of time. I don't know. But yeah, sure, we would like to appreciate if there is a local effect or a circulating effect as we propose, and we are trying to do that by single-cell RNA sequencing, of course, of infected lung to capture how megakaryocytes are actually working. And if I would like to be honest, we are also trying to see if something occurs in bone marrow, so in the production of platelets from megakaryocyte directly from the hematopoietic stem cell, hematopoietic factory. Therefore, we would like to combine these two platelet production mechanism, let's say, to understand that actually what's happening here, even if irrespectively to resolving, I mean, just for the biological point of view.
Can I just ask one last question? So resolvins have been very much a holy grail sort of target in a lot of therapeutic areas. Do you think the perfect drug is going to be giving someone a resolvin compound, or do you think you'd be able to boost endogenous resolvin production? Because that would seem to be perhaps a better
I guess
course?
that maybe the more simple thing is to give resolvin as a drug. I mean, because resolvin is produced by a transcellular mechanisms involving different cell types. So boosting their own production could be difficult because you need to manipulate different cell types. As drug, there are some companies that are working to promote the development of resolvins as drug because these are lipids that were quickly metabolized in cells, so their half-life is very low. Therefore, an advancement for the resolving field, for the resolution field, could be to produce some stable analogs that more potently stimulate the resolution of inflammation. I don't know if I answered to your question.
Yeah, that's great. Okay, I think we'll move on to our next talk. Thank you.
Thank you.
Our next speaker is Dr. Dan Arigides. He'll correct me, is an assistant professor at Dartmouth Hitchcock and the associate director of the Adult CF Center. His work focuses on interactions between macrophages and pathogens in CF, with an emphasis on how modulators affect these. The title of his talk is TSLP and IL-33 alarm and cytokine production by primary human CF lung macrophages induced by infection with Pseudomonas and further increased by CFTR modulators. Thank you.
All right, thank you so much for the introduction and for the invitation to share some of my work here, and I apologize for the length of my title. I have no disclosures related to this proposal or to this presentation. I'd like to start out by positing something and saying that, you know, asthma is common in CF. Up to 30% of people with CF have concomitant asthma, and it's probably higher than that in adults. But we don't really know the full numbers of it because diagnosis of asthma in CF can be challenging due to significant overlap in terms of the signs and symptoms of the disease and the lack of a gold standard for diagnosis of asthma in someone who has a concomitant sort of other muco-obstructive lung disease. And for a lot of those reasons, the optimal management of asthma is uncertain. That being said, asthma treatments are also commonly used in CF, including inhaled corticosteroids, inhaled bronchodilators, and oral corticosteroids, and biologic treatments, including monoclonal antibodies against specific asthma cytokines, are increasingly used, mostly sort of in an anecdotal fashion. At the same time, asthma treatments have the potential to be harmful, and I think that it merits a little bit of a better understanding of what we're doing with these treatments. We know that inhaled corticosteroids can increase your infection risk for non-tuberculous mycobacterial disease. Chronic azithromycin has the potential to induce NTM resistance to this drug, as well as causing some cardiac side effects. Oral corticosteroids recently had, part of the STOP platform investigate this as part of treatment of CF exacerbations and had a negative result there. And they carry a risk for diabetes, worsening control, osteoporosis, and other infections. Beta agonists unopposed have been shown to have an increased asthma exacerbation risk. This was historically thought to be mostly with the long-acting beta agonists, but recently the asthma guidelines have changed to recommend against the use of unopposed short-acting beta agonists regularly as well, due to increased asthma exacerbation risks. And we don't really know what the long-term side effects of some of the biologics are. They generally seem to be pretty well tolerated, but I think it may remain to be seen if there are any kind of longer-term consequences of those. At the same time, uncontrolled asthma is also a risk factor for worse disease outcomes in CF. We've seen recently from Albin et al. That high eosinophil count is associated with an increased incidence of pulmonary exacerbations of CF. And then back as far as 1988, it was shown that a cohort of patients with a positive methacholine challenge had a faster progression of disease over time compared to those without it. And the methacholine challenge is a way of trying to assess whether or not someone has asthma in the setting of CF for the non-clinicians here. The mechanism for the increased asthma prevalence in CF is uncertain. So we do know that Th2 cytokines are increased in Pseudomonas colonized people with CF, as we saw from this publication from Hartle et al. Back in 2006. IL-4 and IL-13 were increased in Pseudomonas colonized people with CF, but not in those not colonized, and it was correlated with worse lung function depending on the levels of the IL-4 and IL-13. Mechanistically, a new class of cytokines, not new, but newer class of cytokines called alarmins represent a potential link between Pseudomonas and asthma. And these consist of IL-25, TSLP, also known as thymic stromal lymphopoietin, and IL-13. And these are cytokines that are canonically thought to be produced by airway epithelial cells in response to noxious stimuli, including viral infection, bacterial infection, allergen challenge, and chemicals such as cigarette smoke or pollution. And these lead to all kinds of downstream activation of multiple immunologic pathways as seen in this simplified diagram here. Alarmins have been implicated in the pathogenesis of CF. We just saw recently Dan Cook's nice paper in a mouse model of CF, as well as some primary human airway epithelial cells, that CF mice were more primed to produce IL-33 in response to an allergen challenge. And this was modified by CFTR modulators as well and decreased in that setting. Back in the pre-highly effective modulator era, it was shown that IL-33 is elevated in the BAL of people in CF. In this paper from 2016, and also in explanted lungs of people with CF, IL-33 was markedly elevated in the airway epithelium there. The roles of the other alarmins, including TSLP and IL-25, are less sort of studied in the setting of CF. At the same time, alarmins represent an attractive therapeutic target potentially. TSLP already has a monoclonal antibody that's approved for asthma, and there is at least one, and I think two IL-33 antibodies that are currently under development for asthma and COPD. And this one, I don't know how to pronounce it, but I think it's itepekimab, has had some phase 2 trials that already look promising and looks like it has the potential to be approved before too long. So it was said in the introduction that we primarily are interested in macrophages in our lab, so why am I looking at an sort of epithelial-derived cytokine? But we're interested in macrophages because, you know, they represent the first immunologic responders to a lot of pathogens in the lung and have the capacity to coordinate a lot of the downstream immune responses there. And when I was looking at some sort of preliminary data about a year ago, looking at the effect of Pseudomonas infection on monocyte-derived macrophages, we performed bulk RNA sequencing and then subsequent ingenuity pathways analysis of these macrophages and found that in addition to some of the more classical inflammatory pathways that you see here, such as the classical activation and pathogen-induced cytokine storm pathways, we also saw upregulation of Th2, IL-33, and IL-17 pathways in both CF and non-CF macrophages when they were infected with Pseudomonas. So this led me to think, you know, maybe these alarmins are relevant in macrophage response to infection as well. So for this work, I hypothesized that CF lung macrophages are involved in alarmin signaling in response to PA infection. And at the same time, we also know that macrophages respond to ETI. It's been shown by our group and others, including some that are in the audience here, that ETI has, or that CFTR modulators have a significant effect on how macrophages respond to pathogens. And so we also hypothesized that ETI will modify the lung macrophage alarmin production in response to PA. So to address these hypotheses, we used our model system whereby we perform research bronchoscopy on both CF and non-CF subjects. The CF subjects that are recruited for these studies have stable, mild to moderate disease with an FEV1 greater than 50%. All of the subjects that the data are shown in this study are on ETI clinically and have mutations that are eligible for them and have been free of exacerbations for at least 6 weeks prior to the bronchoscopy. So these are, you know, represent just sort of stable CF subjects going about their daily lives that come in voluntarily for a bronchoscopy. We looked at the BAL cytokines derived from these procedures as well as isolating lung macrophages. And then we plate them for 48 hours with or without ETI in vitro, and then have some comparison groups with or without Pseudomonas infection for 2 hours, at which point we wash away the bugs, add antibiotics, and then incubate them from additional 24 hours to sort of allow the macrophages to produce a little bit more of the cytokines and increase the sensitivity of our assays. So the first thing that we saw when we looked just at the BAL contents and performed a cytokine multiplex analysis is that CF-BAL contains more IL-8 when compared to non-CF-BAL. And there were trends towards increased levels of IL-1 alpha, IL-1 beta, TNF that didn't really reach statistical significance in this group. But I think it's important to note that even in this group of relatively stable patients on ETI, there still is evidence of ongoing inflammation, particularly neutrophilic inflammation there. We also looked specifically for IL-33 and TSLP biolysas in this group, and in agreement with the data from the German group from before ETI, we found elevated levels of IL-33 in the BAL compared with non-CF subjects. And again, these are people who are not experiencing any significant disease burden. The TSLP levels trended towards being higher in the non-CF group, but they were generally low, and the differences were not statistically significant. So the next question is, this is BAL, we don't know where those cytokines are coming from. Do the lung macrophages contribute to the cytokine induction? And to do this, again, we used our model system in vitro where we plate the macrophages and then expose them to the various stimuli as seen here. When we looked at the downstream cytokine analyses of these, you can see in the third row there that the Pseudomonas infection does induce kind of a classic pro-inflammatory stimulus signature. Not surprisingly, in the CF lung macrophages, IL-8 was very high, IL-6, TNF alpha, among others. But when we pretreated them with ETI, there was actually a decrease in several of the pro-inflammatory cytokines that were seen here. And to highlight this, I've sort of focused in on a few of them, just showing the individual data. So this was 4 subjects that were for this analysis, so it's a relatively small cohort. But we did see a very high induction of IL-1 beta upon infection with Pseudomonas in the blue bar there, and then a subsequent reduction when we pretreated them with ETI. And this is in contrast to what we've seen with monocyte-derived macrophages when used as a different model system, and others have seen as well, where the ETI really didn't have any significant effect on pro-inflammatory signaling. TNF alpha, there were some trends towards a decrease, but not statistically significant in this cohort. But IL-8 was also statistically significantly decreased by pretreatment with ETI. This is in contrast to some degree to what we saw with the other cytokines, TSLP and IL-33. To just break this down based on the infection alone, we did see that TSLP was induced in all of our subjects by infection with Pseudomonas, as was IL-33, and both of these were statistically significant. But there were, when we pretreated with ETI, where, just to remind you, we saw a decrease in IL-8 and IL-1 beta, that there was, in 4 out of the 5 subjects treated here, there was an increase in TSLP upon pretreatment with ETI, and in 5 out of 5 subjects, there was an increase in IL-33 when we pretreated them with ETI. So stark contrast, and shows kind of that the ETI is not sort of functioning as a sledgehammer type mechanism for these macrophages, but it really is kind of redirecting them into an alternate inflammatory pathway here. Finally, when we looked at bronchial epithelial cells as a comparator, given that these are felt to be sort of the main producers of the alarmin cytokines in vivo, we didn't find a huge induction of TSLP or IL-33 when we looked at CF bronchial epithelial cells, and no significant impact of pretreatment with modulators there. And similarly with non-CF control bronchial epithelial cells, we didn't see a huge effect there. So in summary, we found that IL-33 is elevated in the bronchial alveolar lavage fluid of stable people with CF when compared with non-CF controls. TSLP and IL-33 were secreted in response to PA infection of CF lung macrophages. Pretreatment with ETI decreased IL-1 beta and IL-8 production by CF lung macrophages, but it increased IL-33 and trended towards an increase in TSLP as well. So for future directions, we're going to be stratifying some of the BAL samples based on PA colonization status. I think 2 of the subjects in this cohort were colonized with PA, and they did trend towards having higher levels of IL-33, but we don't have enough to sort of reach any statistical conclusions based on that as yet. We're looking at the transcriptomics of the lung macrophages in response to the infection, as well as modifying effects of the ETI pretreatment. And then the outstanding questions remain, are these responses specific to Pseudomonas versus other bugs? And that, certainly is down the pipeline. We're looking at which components of the Pseudomonas are responsible for these effects. And then finally, there's some suggestion in the literature that in addition to producing alarmins, lung macrophages can actually respond to alarmins as well. So whether or not IL-33 and TSLP blocking antibodies will modify lung macrophage behavior, including downstream cytokine production, phagocytosis, and killing of bacteria. And with that, I'd like to thank my mentor, Ali Asher, who's been phenomenally in terms of support throughout this whole time. Diane Mellinger, who has done a lot of the work that was seen here. Lori Gwilt, our research nurse. Tom Hampton and Lily Taub for assistance with statistical analyses. Andrew Calkins, who performed the cytokine multiplex assays in our core facilities. And then Bruce Stanton, Rick Enelo, and Deb Hogan for ongoing mentorship. And with that, I am happy to take any questions.
Excellent talk. That was very clear. You want to go?
Hi, Dipali from Emory. I have a couple-part question. First is, in the presence and absence of ETI, did you see a difference in the Pseudomonas load? And
In the BAL?
yeah,
So we didn't do those types of experiments. So the plus or minus ETI was in vitro when we looked at the, the downstream responses. So we've definitely seen sort of lower returns of Pseudomonas in our subjects when they're on ETI, when we compare them to kind of like historically before ETI. But I guess, does that answer your question, or?
yeah. Second part is IL-33 is more of a repair, tissue repair marker, right?
It's more of like a damage marker. So when the
okay.
epithelial cells are sort of damaged and they during the setting of like necroptosis, it's released out into the milieu.
But it's more towards anti-inflammatory than inflammatory, leading an inflammatory response, right? No?
I don't believe so. I mean, I'm not
Okay.
an expert in that field, but
Okay.
I think it's all of the trials on it are sort of looking at anti-inflammatory effects of the blocking antibodies and
Right,
trying to decrease COPD exacerbations, asthma exacerbations, et cetera.
Exactly. So jumping off of there, if IL-33 was reduced in the presence of, increased in the presence of PA infection, PA infection will lead to more, there are studies which suggest that more infection of Pseudomonas, there is an upregulation of inflammation simultaneously that's happening in the airways. So
Yep.
if that physiologically is happening, then IL-33 should probably be going down because there's more inflammation. Do you have anything to comment on that? I'm trying to just understand this physiologically.
I mean, IL-33 is canonically more induced by infection rather than repressed by it.
Okay, thank you.
Thank you very much. There's quite a lot of evidence, especially in vivo, that type 1 responses are favorable in CF, right? So I was wondering whether, in your experiments, whether you had a chance to add some type 1 cytokines, in particular interferon gamma, in conjunction with the ETI to see if you actually improve even better the upregulation, or you restore basically your phenotype with a type 1 type of mediators. Do you have a chance to do that?
No, I haven't done that. That would certainly be interesting, and I think there's a lot of work on sort of like the balance of antiviral and antibacterial immunity, and Jen Bomberger has done a lot of that work at Dartmouth. Certainly interesting kind of proposition for future experiments, but I haven't had a chance to do that yet. Yeah.
I think that'd be worthwhile.
Yeah, thank you.
Hi, Carla Ribeiro from UNC Chapel Hill. I appreciated your talk. I have a comment and then a question.
Sure.
The comment is we also have shown utilizing a different approach, which is usually our traditional model in the lab, exposing airway epithelial cells in primary cultures, HBs, to supernatant from milk or purulent material harvested from excised CF lungs, or alternatively, to expose the HBs to BALF from a pediatric cohort. And in either way, we of course trigger a lot of inflammation, but we showed that there is no effect of ETI on decreasing the inflammatory responses of either induced by BALF or by what we call SMM. So my question to you is, what would be, in your view, the differences? Because you also have shown that the HBs do not, respond to ETI. What is the difference that you can envision between a lung macrophage versus a human bronchial airway epithelia in terms of how they respond to ETI from an anti-inflammatory effect?
Yeah, I mean, that's a really interesting question. I mean, I think we're obviously a little bit biased towards the macrophages and in sort of thinking about how they coordinate the immune response. As you know, it's somewhat controversial whether or not the macrophages have CFTR on them. I don't think this is a surprise, but we've looked at sort of how they respond to the ETI and whether or not it's via a direct effect on CFTR or via sort of off-target effects. We don't have the data to sort of support that yet. I know Ben has sort of done some of that work looking at the effects on the chloride channel conductance on the macrophages. I think the epithelial cells and the macrophages are really just sort of fundamentally different cells in terms of their role in the immune response. Obviously, the epithelial cells have a role to play, but their primary function, if you may, would be to sort of be the physical barrier and sort of be part of the airway lumen and sort of make that barrier on there, and they respond in that setting, whereas the macrophages are really the cells that are there primarily to orchestrate immune responses. So they're probably more primed, if you would, to have an inflammatory response to a stimulus at baseline, and that may impact their downstream responses to the modulators.
Dan, that was a great talk, and I love the resource you guys have to bronch patients. You mentioned you were interested in looking at Pseudomonas positivity and non-positivity. Do you have the numbers to look at what people with different medications on board, and if that is affecting these responses? Because you started off by saying there's a cohort that has a subset that has asthma. So do you know if any of these patients are taking inhaled medications, and are they taking azithromycin, and can you look at that?
We do have that data. We don't have the numbers now to make any sort of conclusions based on that, but I think that would be really fascinating. The problem that you run into is that there are so many different potential medications that I think if you wanted to truly get into like what are the contributions of beta agonists, the contributions of inhaled steroids, the contributions of tobramycin, of all these things, you would probably need a much larger cohort than a single center would be able to provide, unfortunately. But I think that's a fascinating question.
Would you ever be able to take the cells and ex vivo expose them? Do you think that model would work? Or
yeah, I definitely plan to do that down the line. Just sort of in the process of going that direction now. Yeah. Thanks.
Thank you for the great questions, and to the speaker, Dan.
Thank you.
Okay, so we're coming on to our final speaker now, Dr. Spencer Poore, who is an assistant professor and pediatric pulmonologist at the University of Alabama at Birmingham. So He's going to tell us today about elexacaftor, tezacaftor, ivacaftor inducing a Th2 cytokine increase in CF sputum that results in a Th1 to Th2 class switch in airway inflammation. Quite a mouthful, but you can now bring us home, Dr. Brewer.
Well, the caboose is here, so we're ready to go. So thank y'all for having me. I appreciate it. This is going to be, it's not that, physically higher up than what we've talked about, but it's going to feel like a 30,000-foot view compared to the talks before me. So I appreciate everything. Okay, disclaimer, I get some funding from CFF and some loan repayment and things. So as you guys have probably figured out, inflammation is quite complicated in the CF airway. You got this really pretty figure here that's got all sorts of different pathways and arrows and all sorts of different directions, and every single one of these they've gone into great detail with. Well, guess what? There's plenty more figures like that describe very similar things. These look like black-eyed peas to me, but I'm from the South, so that's okay. Pretty complicated situation, very interesting process. Well, I can't talk about inflammation without talking about these cell types too. More epithelium, more other cell physiology. Oh wait, here's another figure that talks about even more mucus and more infection and other things. And another, this is a really good paper, by the way, I like this one a lot. But look, here is even more inflammation, more processes, more arrows, more letters and acronyms, and all sorts of things, making this even more and more complicated. I don't even know what this one's about. I just put it on here. Can't talk about inflammation without talking about macrophages, or people like Katie and all these folks are going to get up here and yell at me. So I had to put a picture of that on there. And then look, oh, here's some more. We got to make sure those are okay. Oh, I did another one. And there's a lot of complexity here. And then there's these beautiful mucus pictures from UNC that look like, the backdrop to the Dead and Company Sphere residence in Vegas right now, which is great. But you're probably looking at me and thinking this as I just threw all of those things up there. And that I'm a few minutes away from lunch, that's probably even more evident. But what I'm going to do is I'm going to try to make this a little simpler, okay? And we're going to start simple with just that this neutrophil, Th1 inflammation in CF is complicated. And I'm coming at this from a very clinical perspective of that what we're going to talk about. So certainly there's strong outcomes correlation and things that Dan just talked about. A lot of associations with IL-8 in the sputum and airway showing decreases in FEV1, like kind of our biggest clinical markers that we have. Certainly responses to infection and injury. It's not necessarily the first response, but it's not the last. Certainly high concentrations of these cytokines in CF sputum across the board. And we see pretty dramatic changes during what we call pulmonary exacerbations in this type of pathway. This is a really great figure that kind of shows that imbalance that we classically, I haven't been around that long as you can tell, that we talk about when we think about how inflammation of the airway results in a systemic response in people with CF, as well as what specific processes may be playing a role with the neutrophil, and how you have these T2 cells that may have a shift in a dynamic change. However, people have looked at this before, as Dan 1 has talked about, and Dan 2 in the back wrote a paper about, is that there is Th2 inflammation in CF is also complicated. People have shown for years there are higher rates of allergic rhinitis and nasal polyposis in this patient population. Aeroallergen reactions, highest mucus burden with like an allergen entrapment that you would kind of see, similar to bacteria. High rates of ABPA or ABPM, or mycosis is how they would describe it. Higher, like Dan just said, higher IL-4 rates in certain airway samples with chronic Pseudomonas infection, okay? And association with specific mucin type, CFTR function, even epithelial cell signaling, okay? Now, to prove it, these are some papers from back in the day where they were talking about this type of condition, pathways in CF. They were, this is a few dinosaurs were still around when this was going on, but a lot of what was happening is 1961, the Schwachman, that the award's named after, here in Boston itself, okay? This isn't the first time that people have asked this question, but with ETI now being present, it's really important that we revisit it and think about it hard. Segueing to that, a lot of people, and many of them are in this room, have looked at this exact situation. How has ETI altered this inflammatory response? So, certainly we've seen notable changes pre and post ETI, changes in FEV1, hospital admissions, mucus content and the character of that mucus, quality of life metrics, as well as little changes, however, in people with CF and ABPA rates since 2014, which is a little odd. You know, the lowest that the highest that it's been is 5% since then, and the lowest it's got is about 4.5%. So it hasn't changed much despite these modulators. And we all know that people with CF are still having symptoms. Now they're changing and it's different. Nobody can deny that, but it hasn't been eliminated. And we've certainly suspected a Th1 change in this neutrophil-driven process of that weighted scale that I showed you before. But this Th2 allergy component, IL-4, all these other aspects, how is this changing? How is this adjusting to this new world? So I ain't gonna lie. This guy over here, who some of you probably know, has many talks that he's associated with looking at this, okay? So this is Dr. Sagal, who's one of my mentors in fellowship and one that has certainly helped me with this particular project. So I'm gonna kind of take my little pepperoni on the promised pie, I guess, as you could say here, and talk about my specific part, but I wanna say that there are a lot of talks and a lot of posters and a lot of sessions that have been going on specifically asking and answering this question, okay? So kudos to them. They've helped me do this one. So let's get right into it. So this study that we did was based very simply off PROMIS study markers that were already being run through the CFF biomarker resource lab that Dr. Sagal runs in Colorado. He was very interested in changes in neutrophil activity and neutrophil markers in the sputum of people with CF before and after they started ETI. It's a big, huge PROMIS study initiative, okay? Well, I happened to be a fellow there at one point in time, and I said, well, I'm really interested in this. I mostly look at fungus and aspergillus, and these are some other aspects and cytokines that have come up, and can we add them on? And he said, sure, no problem. So what we did is we certainly correlated and associated this with many of the other markers he was studying, as well as inflammation and other outcome measures. And certainly I was just going to tell you that this is an ongoing analysis. There are certainly other markers and things coming down the gate. But from an equity standpoint, a lot of it is with the CFF biorepository, so we were working closely with them. Here, this figure on the right, I'm not going to go into great detail, table, sorry, kind of talks about the overall cohort as well as the specific sputum cohort. A lot of this was used for pretty basic demographics, nothing that was too abnormal or too outside of kind of what we have seen in the population over the past few years. But as you can see here, if you're interested, there's certainly genotype breakdown as well as historically these types of pathogens individuals had and whether they were on modulators or not prior. So I was specifically looking at IL-4, IL-5, and IL-13. So the first thing we did with the help of Brandy Wagner, she's a statistician out in Colorado who is out driving a lot of this, is we looked at said, is there any baseline measurements, any markers of these three that show any correlation with anything? Not much, I'll be honest with you. You don't have to read it too much. But most things here, there wasn't a great correlation necessarily with age, FEV1, Pseudomonas, whether it's currently infected, BMI, lower airway disease, because there is a population of these that are pediatric, as well as quality of life or sweat chloride at the baseline visit prior to starting ETI. However, this is basic means looking at these values across a 1-year period following ETI administration. So these are sputum samples obtained from the PROMIS study of people who in the pre-phase were not on ETI and then subsequently started it and continued through the actual PROMIS study. As you can see here, that IL-4 and IL-13 certainly had some pretty impressive responses, but they did kind of vary. There wasn't a straight plateau, which this is more of Scott's information and things, so I defer you to his, because this goes in much detail, this is what IL-1 beta and IL-8 were showing in our PROMIS studies, as well as some of our CRP, which is our one systemic marker I've kind of looked at right now. These are all airway markers. So a much different pattern between these two. So what we do with any clinical study or anything, we test it and push it a little bit further, control for more things, look for more confounders, and try to push that system. So that's what we did. Here we started to pair things and we cleaned up the data a little bit. We started to say, okay, you had to have 2 markers, you had to have certain number of visits and things, and we found that this trend still persisted, okay? We still saw this increase at 1 month, and then a notable one at 12 months, looking at these adjusted markers in the sputum. So we then took that and said, okay, well, let's look at 2 of these together. IL-13 seemed to have a more dynamic response. IL-1 beta kind of hit a little bit more spaces, pro-inflammatory. You got a little TH1, a little TH17, a little bit of more there. And so you had to require at least 2 values each, okay, a measurement of some sort, because as you know, with clinical data, it was a decent spattering of things. Pretty much all the subjects in this little matched cohort, they all had some IL-1 beta response that we could measure, okay? Interestingly, about 23% of the subjects didn't have an IL-13 response at all, but did have an IL-1 beta response. And that's what this is shown over here to the right. Now, when we correlated these 2 together, putting them side by side, it was very nice that Dan was speaking right before me, because you got to see some of this changes in this difference. So we did some slope analysis and things, and the slope is certainly not 0 in each of these situations, and it was statistically different between the 2, showing that there was an increase over time in IL-13 as well as a decrease in IL-1 beta. So what do we do with this again? We push it even harder. Okay, so now we took it out to 2 years, and we did a multivariate analysis, okay, specifically looking at IL-4 and IL-13. And here what we did is we started to control for age, previous modulator therapy, okay? And then we also did a fancy thing that I don't even know how you do it, where Brandy was able to take those 0 or unmeasured values or offset below the detection limit and was able to apply a random number within that off-limit range. So really strengthened this. And interesting, and we also took it out to 2 years. That was data that they had available. We saw very similar findings in IL-4 and IL-13, but then we saw this odd drop again. Back down at 2 years, we saw this change, similar to how they were at 3 and 6 months. We then took visit 5, so that 12, so that would be that peak of those, and then this visit 6, which was the 2-year, and the drop, and still didn't see a lot of correlations or anything with any outcome measures. Of note, we don't have sweat collide for this one, but we do have it for the 2-year mark. So there wasn't a lot of big differences from those outcome measures that we traditionally have been using. So in basic summary, following ETI initiation, the Th2 cytokines rose in the sputum as Th1 dropped, as so did the systemic inflammatory markers. This was sustained after a year. However, at 2 years, there was a reduction again. Now, these similar drops were seen at 3 and 6 months of unknown etiology, and our advanced multivariate analysis certainly showed similar results, but there weren't any associations with any other outcome measures that we could find. So what does this mean? Okay, well, I'm going to cheat and just say that it means that inflammation in CF is dynamic and obviously changing with ETI, okay? Th2 cytokines are increasing. There's a lot of work being done by some very smart people in this room that have shown that there is a lot of changes going on that we don't fully understand. Why are they fluctuating over time is not clear and certainly needs further investigation. As you've seen from all the talks before me, wherever you're modulating CFTR has a chance to change what the host and the inflammatory response can occur. Clinical observations and symptom monitoring is also needed to better understand how it's altering this disease, okay? And then I like this picture over to the right that was in Trends in Immunology that's actually more representative of an asthma picture, but I think it shows how it sometimes feels right now, in a good way, when we're thinking about individuals with CF, is that how are we shifting and changing this inflammatory phenotype in the airway? Are we making more of an eosinophilic or a neutrophilic change? Are we moving it towards what one would consider closer to normal or baseline? But we don't really know. Certainly we need a lot more work and a lot more effort to study the basics of the cells as well as much more clinical outcome data. So I'm going to pause there because I'm done. And so certainly thank you to Scott Sagel in particular, as well as Brandy, who really, when you say you're standing on shoulders of giants, I'm certainly doing that right now, and I really appreciate it. Steve has been very helpful with this as well. as well as Jing at the TDN. I guess I did want to throw out one thing to a lot of people that, it's pretty impressive with this foundation in this situation where I was able to put this presentation together, write abstract, and base, lots of talks off of, and I haven't even touched one of these samples personally myself. I guess I technically did in fellowship because Scott made me learn how to process sputum. It's not fun, but I wanted to purposely thank, certainly the Promise leaders, all the Promise site investigators out there, and certainly the Promise research staff, this is our crew over here to the right, a lot of people that actually interface with these individuals, are able to get these samples under the face of a lot of what we do, and certainly the Promise participants, because if it wasn't for people with CF, we wouldn't even be asking these questions or thinking about these things. So if you wanted any references for all those crazy pictures, there they are. And I'm happy to take any questions before your lunch.
Thank you, Spencer. That was a great talk delivered with Southern style. We liked it.
Thank you. Appreciate it.
So could I just ask a quick question before we go to the floor? So do you have any inkling of whether there might be environmental exposure being involved in changes in the cytokines in these patients? So time of year, allergen exposure, etc.?
That was the first thing that my gut wanted to say and look at. However, dates and timing and of how this was run was spattered through time and time and time. And so to really get granular with that would be really difficult. But that's kind of what, you can't ignore that could happen, but we need to really think about the dates and the timing and the tracking of these to really understand.
I was going to ask the same question because it looks like you're starting to get seasonality in here. So actually collecting when, putting that, forcing month of collection into the model, I think is going to be really important, because now that you don't have an airway that's dominated by huge numbers of bacteria, the nasty things that cause Th2 inflammation, like rhinovirus, becomes a really big thing.
Exactly. And so I think that is kind of, what other situation do you get a similar response 1 year later? So we really need to focus on timing, dates, and what are the pattern that these individuals were experiencing when we took these samples. That's great.
Hi, Thomas. Lovely talk. My name is Nicole. I'm from Dartmouth College. I was actually just curious. So we've had a lot of talks also showing that ETI has impacts on the microorganisms that are present in the CF lung. And with your talk showing that you had an overall trend of an increase in more allergic-like responses, but a decrease in Th1 responses, do you think that ETI treatment long-term is kind of promoting colonization and constant allergy insults, or do you have any idea of how this is impacting the microbiome and subsequently the host environment?
Yeah, no, that's a great question. So I don't know, frankly. It's pretty evident that there has been some dramatic shifts, okay? Certainly, I look a lot at aspergillus and things, and certainly people would say, aspergillus and starting to thrive in IL-4 associated environments and these kind of things, but that's not what the data shows, okay? Aspergillus tanked, okay? It dropped way off. They just released that paper in Blue Journal a few days ago, okay? And to say exactly how it's going to affect, now we're certainly looking at that, that's part of this, okay? Are there thoughts behind when you take, like Ron was talking about, when you take a bacterial load and an inflammatory response and shift it, does that change the real estate? Does that change an exposure? Does that wake cells up in a certain way? Does that motivate things? I think that's where we have to get a lot more granular look at how the response is changing in each one of these conditions.
Thank you.
Okay, well, I think we'd like to thank the speakers and then also,